So the nervous system is going to be composed of really two groups of cells. We have neurons, and then we have neuroglia cells. Neurons are really the main cell of the nervous system. So neurons are those excitable cells that can rapidly change their resting membrane potential or their voltage across the plasma membrane in order to send or receive nerve impulses. So neurons are excitable cells, which means that they can rapidly change their resting membrane potential, or their RMP, or the voltage across the plasma membrane in order to send and receive nerve impulses. The second group of cells in the nervous system are neuroglia cells. Neuroglia cells are known as supporting cells. So neuroglia cells are non-excitable, which means that they're not changing that resting membrane potential or sending and receiving nerve impulses. Instead, the neuroglia cells are supporting those neurons. So neuroglia cells or glia cells are known as supporting cells. They're non-excitable cells, which means that they are not sending or receiving nerve impulses. Instead, they're just supporting our excitable cells in the nervous system, which are our neurons. 99% of the cells in our central nervous system are actually supporting cells. So the central nervous system, remember, is going to be the brain and the spinal cord. The majority of the brain and the spinal cord are going to be composed of neuroglia cells. However, it's also important to realize, though, that 50% of the brain by volume is actually going to be composed of neurons. So first, we're going to focus on our neuroglia cells. There are 6 different types of neuroglia cells. There's 4 types of neuroglia cells in the central nervous system, and then there's 2 in the peripheral nervous system. So A through D is going to showcase the neuroglia cells in the central nervous system. So we have astrocytes, microglia cells, ependymal cells, and oligodendrocytes. Those are going to be the neuroglia cells of the central nervous system. And then in the peripheral nervous system, we're going to have satellite cells and Schwann cells. So satellite cells and Schwann cells are going to be the cells of the peripheral nervous system. Oligodendrocytes, ependymal cells, microglial cells, and astrocytes are then going to be the cells of our central nervous system. So first we'll look at our central nervous system neuroglia cells. The first one being astrocytes. So astrocytes are these kind of star-shaped cells. And astrocytes have these protrusions, or these appendages that reach out and wrap around capillaries and neurons. So astrocytes are providing some physical support to our neurons. You can see that they're wrapping around those neurons and grabbing on. And they're also surrounding these capillaries. This allows for astrocytes to not only provide physical support to our neurons, but also really keep that nutrient supply very close to our neurons. So neurons are really active cells. They're sending and receiving electrical impulses over and over again. So they're going to need a lot of nutrients. So astrocytes are going to be very helpful in keeping this capillary close so that there can be an exchange of nutrients and waste. Additionally, astrocytes are going to monitor the extracellular fluid, so the fluid that surrounds the neurons, and it's going to pick up any excess neurotransmitter or ions and then bring it into the astrocyte. So it's going to help to recycle ions and neurotransmitters from that extracellular fluid. So astrocytes are going to function to provide physical support by grabbing onto the neuron. They're going to keep the nutrient supply close by grabbing onto a capillary. and they're going to control the contents of the extracellular fluid surrounding a neuron by recycling any excess neurotransmitters or ions. The second central nervous system neuroglia cell is the microglia cells. Microglia cells, as you can see here, are going to have these kind of thorn-like protrusions. These kind of thorny protrusions or thorny processes are going to reach out, and they're actually going to touch nearby neurons. Microglia cells are really monitoring the health of nearby neurons. So they're reaching out in order to monitor the health of this nearby neuron. If for whatever reason this nearby neuron was injured, so for example, maybe this neuron has now been injured and there's some cellular debris around, this microglia cell can actually transform into a macrophage or a phagocytic cell. Macrophages participate in phagocytosis, in which case they can actually engulf and get rid of neuronal debris. So again, if this nearby neuron was injured, this microglia cell has this thorny process that reaches out and touches that neuron. It's monitoring the health of nearby neurons. So if this guy was damaged and there was any neuronal debris, this microglial cell could then convert into a macrophage-like cell, and then it can actually phagocytize any neuronal debris. Additionally, microglia cells are going to be monitoring kind of some of that surrounding fluid, watching for any foreign microorganisms as well. So if there were any foreign microorganisms around these neurons or infecting these neurons, that microglia cell could once again act like a macrophage and engulf that microorganism. The third type of neuroglia cell in the central nervous system is an ependymal cell. Ependymal cells we're going to talk a lot more about when we cover the brain, but for right now, I want you to know that ependymal cells are going to actually line these fluid-filled cavities in the brain called ventricles. So ependymal cells are lining these fluid-filled cavities called ventricles. Ependymal cells are going to help produce the cerebrospinal fluid that then fills these cavities. And ependymal cells are going to have abundant cilia that are then going to help to keep that cerebrospinal fluid moving through those fluid-filled cavities or those ventricles. So ependymal cells line the ventricles in the brain. Ventricles are fluid-filled cavities. They also help produce the cerebrospinal fluid, and that's the fluid that fills these cavities. And the ependymal cells have cilia that can then whip back and forth to keep that fluid or that cerebrospinal fluid moving. Our last type of neuroglia cell in the central nervous system is an oligodendrocyte. Oligodendrocytes have these arms or appendages or processes that extend out, and they're actually going to wrap around the nerve fibers or the axons of neurons. So oligodendrocytes have these processes or appendages that extend out, and they're going to wrap around the axons of our neurons. This is going to produce what's called a myelin sheath. And a myelin sheath is really just an insulating covering around the axons or around those nerve fibers. So oligodendrocytes are going to form myelin sheaths or those insulating coverings around the axons. In the peripheral nervous system, we have two types of neuroglia cells. We have satellite cells, and we have Schwann cells. Satellite cells are going to surround our neuron cell bodies, and they're going to play a similar role as our astrocytes. So astrocytes are a part of the central nervous system. So they're one of our neuroglia cells in the CNS. They function to provide some physical support. They help to control the extracellular fluid around those neurons. And they also are going to help to keep that capillary or nutrient supply close. So satellite cells have similar functions as our astrocytes in the CNS. Our second neuroglia cell in the peripheral nervous system is going to be a Schwann cell. Schwann cells are functionally similar to our oligodendrocytes in that they're producing that insulated covering or the myelin sheath that's wrapping around the axons of our neurons. What you'll notice is that Schwann cells are wrapping their entire cell around that axon, whereas in oligodendrocytes, you'll notice that the oligodendrocyte cell body is here, and then it's just actually one of the processes of the oligodendrocytes that's wrapping around an axon. Whereas a Schwann cell is wrapping that entire cell around that axon. Schwann cells are also going to be extremely important in the regeneration of any damaged peripheral nerve fibers. So Schwann cells are going to actually help to regenerate any damaged axons in our peripheral nervous system. As a part of their role in regeneration, they also are helping to pick up any damaged or any neuronal debris. And so they can also be phagocytic and engulf any cellular debris associated with that site of injury.